In this section, we're going to discuss the continued part of the nervous system, but this time through more physiology rather than histology of the nervous system itself. And this segment is dedicated to our studies of action potential. So before we go into completely new material, let us just put back on the same track where we left while we were discussing nervous tissue as part of the human body, one of the four types of tissues that are observed. And this diagram has already been used. By doing this, we identified that the nervous system composed of neurons, of course, shows some kind of color differences depending which part we observe, whether it's a cross-section through the spinal cord or cross-section through cerebral hemispheres. We identified two different types, the white matter and the gray matter of the central nervous system. We know right away that where we see slightly darker coloration of brain or spinal cord tissue, the gray matter, we immediately draw the conclusion that it is more heavily populated by bodies of neurons and dendrites. And of course, there should be some axons, but number of axons that are found within the gray matter is mostly unmyelinated axons, because the color difference really comes from the myelin coat added by Schwann cells for the peripheral nervous system, but oligodendrocyte cells in the central nervous system. Obviously, the white matter, whether it's spinal cord or brain, will be area or part of the organ structure that we study, where dominance of myelinated axons will be something that we make as conclusion. Of course, we need to remind ourselves also about some basic things that we covered when we discussed the individual nerve cell, neuron, or nerve fiber. On the top of the diagram, we see that it is the body of the cell containing all the different structures, organelles, nucleus, like any other living cell should have. However, there was immediately one huge difference, which made neuron uncomparable to any other cells. Presence of these projections that are collectively known as dendrites, and they're quite massive, branching off, creating dendritic spines, where each and every of the ends that we can see enables this neuron to communicate through the synapse with other neurons of the central nervous system. And of course, the language of communication is what we define later on as a synapse. And of course, when they communicate the language that neurons are utilizing to interact between themselves, as well as with other tissues, is the electrical current that we call action potential, that we call nerve signal or nerve impulse or nerve stimulus. These are all same things that we interchangeably use. So once this neuron becomes stimulated by other neurons, essentially it would start gathering electrical charge throughout the cell. And as a result of the sum of all received charges, basically, it is the axon hillock, this slightly enlarged part of where the axon is attached to the cellular body. This is what we call the axon hillock. Here's the label. At that point, neuron decides to generate its own electrical charge, signal, or impulse, and to send it down its axon. Keep in mind, because of certain changes that would affect the cell which is generating its own action potential, electrical current cannot propagate backwards. It could be only sent one direction along the length of axon from the cellular body axon hillock all the way down to the end, which is axon terminal and its synaptic end bulbs. The axon is illustrated here quite properly with some kind of sideways directed branches, which means that axon is going to influence more than one cell. Either it's going to do through multiple collateral branches, or at its very end, or at the end of each individual branches, it'll form axon terminal with slightly distended ends that are called synaptic end bulbs. And this is something that we can address right now. Within those synaptic end bulbs, individual nerve cells accumulate and store substances, chemical substances that are collectively known as neurotransmitters. Release of neurotransmitter substance from this neuron, which is called the presynaptic neuron, is going to start affecting cells, other neurons that are functionally defined as the postsynaptic, because the neurotransmitter substance will start influencing the postsynaptic neurons to start changing polarity of their own membrane. And as a result of that, eventually would force them to generate their own action potential, electrical charge, signal impulse, which would travel along the axon until it reaches the effector cells, whether it's a gland, whether it's a muscle, stimulating them to fulfill their own action. So in order to make sure that we're absolutely on the same page, we just need to, again, remind ourselves of presence of different channels. We talked about them earlier in the cytology section, where we talked about cellular membrane and then proteins that are integrated into cellular membrane. For some of them, we identified them right away that they serve as ion channels. And at that point, we have made also a very direct statement saying that ion channels could not be seen as general structures that will allow any ion to pass through. They are ion specific. So as we see here on the top part of the screen, this protein channel is only to allow movement of potassium ions. What is in the background? In the background, we talked about different concentration gradient of ions across both sides of cellular membrane, but we also talked about the electrical gradient, saying that the sum of all charged particles on two different sides of the membrane generates what is called the potential. So slight difference in presence of charged particles on both sides of the membrane generates the membrane electrical potential. And as such, it is about negative 70 millivolts for this cell in its resting state. So what's going to force this channel to open up? Basically, there are three different possibilities. One of them, which is not illustrated on this diagram, and I'd like to start, is that some cells would respond to physical stimulus. For example, many of these receptors that we have within our skin, they respond to some kind of physical changes as we press our fingertip against something that we're trying to explore. That is going to be enough stimulus to start opening some of the protein channels and to allow movement of ions into the cells, generating action potential on the basis of this kind of physical deformation of their own membrane. So that's the physical. However, it could be also with change in the voltage of the cellular membrane, or at the bottom part of the screen, we can talk about chemical stimulus, practically something that we can say any substance that is influencing another cell. Potentially, we call it ligand, and these are ligand-gated channels. Okay, let's see and let's explain what's going on here. With some change affecting the membrane of the cell, it starts changing its resting membrane potential, changing the voltage, moving it from negative 70 towards higher levels. And in this case, to the right, we see that the voltage has risen to negative 50 millivolts. That would be sufficient change in the membrane potential that would influence this protein channel to open up. So before it, the gate was closed. When the voltage is changed, the gate opens. And then what happens? Basically, ions, in this case potassium ions, just flow down their own concentration gradient from where they're present in a higher concentration intracellular fluid compartment in the cytosol into extracellular fluid compartment. So diffusion of potassium ions is going to be observed, but in order for this to happen, basically potassium channels must be open. When the stimulus is gone, basically voltage is going to go back to negative 70 millivolts. The gate would close and further movement of potassium ions will be eliminated. We may have a protein channel that doesn't respond to electrical changes, but rather responds to presence of a specific molecule, which it wants to bind on its receptor site, on its surface. That molecule is what we call the ligand. And in this case, we can say this might be idea of this is neurotransmitter substance, which when binds to protein channel, actually it's going to make it open. If the ligand is not within a receptor site, the protein channel remains indifferent and stays closed. So we can see here that binding a ligand forces the channel to open. And for as long as the ligand is present, it is not decomposed or it doesn't drift away. Basically, the ligand-gated channel is open. And you can see here that it is going to allow movement of substances. Practically, Illustrator opted for moving them both direction in and out of the cell. But here the point is to understand what's the difference between the voltage-gated channel versus the ligand-gated channel, not necessarily just to kind of take it as is and to say, well, voltage-gated channels allow transport both ways. It is only for illustration purposes. If there is any kind of dilemma, this is what you and I were already talking about. Extracellular fluid containing positively and negatively charged ions. Same situation applies to cytosol or intracellular fluid. So basically, we know that we're not completely electrified because the sum of charged particles appears to be very close to equal. And of course, extracellular fluid is neutral by itself. Intracellular fluid is also neutral. But because of slight difference between positively and negatively charged particles on both sides of the membrane, basically this is hypothetical idea that we keep using for as long as we need to keep explaining things such as action potential, physiology, or contraction of a muscle. So for that reason, most of the time you're going to see positively charged outside part of the membrane and the negative charge being added or illustrated to the inside of the cellular membrane. So keep this as some kind of just a basic idea to help you understand, not to say extracellular fluid has positively charged particles while intracellular fluid only houses negatively charged particles. That's not correct way of seeing it. We're just talking about minute gradient between positive and negative charged particles just directly and immediately next to both different sides of the cellular membrane. So if we would insert one probe into a cytosol and we would keep another probe of a voltmeter on the surface of the cell, reading would show that we're reading negative 70 millivolts, and that is what we consider to be the cellular resting membrane potential. Resting because whatever we do, it should stay like this throughout the length of our life. But with some changes, of course, that kind of charge starts changing. And as it changes, basically, it measures a little different than just having a flat line of negative 70 millivolts. So disregard for a moment the titles hyperpolarizing and depolarizing. I will explain that later on. But this is what happens. If certain stimulus has been applied to the cell and it affected distribution of ions next to the cellular membrane in a form that voltmeter starts reading 72, 75, or closer to negative 80 millivolts value. Resting membrane potential is actually creating this form of indentation, little dip, which would make it, interestingly enough, a little bit harder to stimulate cell to generate its own action potential, while in this red colored part of measurements. However, a similar stimulus being applied to the cell, which changes resting membrane potential, taking it to 68, 65, closer to negative 60 millivolts, obviously changes membrane potential at rest, but in the opposite direction. We're calling them hyperpolarizing and depolarizing graded potentials. For both of them, it is going to be interesting. For hyperpolarizing and depolarizing, this cell will remain neutral, indifferent. It won't generate its own action potential because the changes that affected part of its membrane were insufficient to trigger another action potential to be generated by a cell. Hyperpolarizing made it even less likely. Depolarizing potential made it more likely, but not enough to trigger it. So we're going to see this, and I like using this slide just for introduction of terms such as depolarization, hyperpolarization, and this will be explained a little bit further when we take a look at this diagram, which will take us through the entire cycle of what generates action potential. So let's see it here. In a lilac-colored part of the diagram, the voltage is measured negative 70 millivolts in a constant form. But at this point, between lilac and light blue, we obviously have some kind of stimulus being applied to the cell. Doesn't matter what kind of stimulus. It could be physical, chemical, electrical. It will be, at the end, all the same. So as the stimulus started changing the polarity of the membrane, it is allowing it to go up, and if it stops, it's going to come down to zero. But if the stimulus was strong enough to generate sufficient changes to bring the membrane of the cell to negative 55 millivolts, that value is considered to be the threshold. So what's going to happen? We were opening some ion channels as a consequence of stimulus being applied to the cell. Therefore, movement of ions across the cellular membrane started changing polarity of the cellular membrane, taking it all the way up to negative 55. At this point, negative 55, it's considered to be already significant change. That's why we have the value or idea of reaching the threshold. At that point, the rest of the voltage-gated channels start opening very quickly, almost all of them simultaneously. So with additional movement of ions across the cellular membrane, now we can see here that this cell actually has a very steep and quick change in the polarity, going from negative 55 to 0 to plus 30. And that part of the action potential is what we call the depolarizing phase. When the depolarizing phase reaches its peak, at that point, certain channels do not have any further incentive to stay open. They, quite the opposite, shut themselves closed. But we're entering the red-colored part of this diagram, which is called the repolarization phase. We see what's happening here. Polarity of the cell, which has changed from negative 70 to plus 30, now needs to be reversed. We want to bring this cell back to negative 70 millivolts to make it ready to do the same change when the next stimulus is applied to this cell. So to keep doing this repetitively. And of course, in order to bring the polarity back to 0 and back to -55 and -70, we would need to think about, could it be done by same channels that we now, again, reopen them? No, it would not be good enough. We have to opt for different channels that allow different chemical compounds, different ions to move out of the cellular space, out of cytosol. So if you don't mind, I would just remind you of this by adding arrow in, and I will remind you by putting ions are moving in to the cell. And we know that for this part, it's going to be mostly due to sodium single positive charged ions that would be part of the game that we're describing practically as the depolarizing phase. Sodium ions quickly move in. In a repolarization phase, we have to have those positively charged particles being moved out. And moving them out certainly cannot be sodium. It would be ideal, but it's not like that in nature. Moving out will be seen as movement, massive movement of potassium ions through their own channels, and they are coming out of the cytosol. As they come out, of course, they carry the same positive charge, and therefore we're seeing positive charged particles. So sodium went in, it changed. Now we're letting some positively charged particles getting out of the cell. It is going to change and will bring us back to resting membrane potential. With potassium channels, they cannot close momentarily when we hit -70 value being here. -70 value is not the end, because they are relatively inert closing, will allow a little bit more potassium to keep coming out of the cell and would change the membrane potential to become less than -70, maybe -72, 74, 75. Doesn't matter the absolute value. What matters is that we understand that when we go further into negative territory below -70, basically this part of membrane potential is known as the after hyperpolarizing phase. And at one point, certainly we're going to allow the cell to come back to normal, to complete its repolarizing phase, and practically be ready at -70 millivolts for another cycle of the same kind, generating another signal, another action potential. So let's take a look and see what the legend is. For the purple colored part, or lilac, resting membrane potential, voltage gated sodium channels are in a resting state, and voltage gated potassium channels are closed. So sodium, potassium are kept on both sides of the membrane, no movement across the membrane, therefore practically quite steady resting membrane potential. The stimulus being applied practically is leading to depolarization. Based on quality of stimulus, repetitiveness of stimulus, basically we can see that either it resulted in short but incomplete depolarization, or it leads to more complete depolarization, taking us to the threshold level of -55 millivolts. The green colored part of this diagram, referring to depolarization, practically confirms what I tried to already introduce. It's the voltage-gated sodium channels that become open, and this cell very quickly changes its membrane potential, going all the way up to +30. But when it enters the repolarization phase, it is voltage-gated potassium channels that are open while sodium channels are inactivated. So sodium came in and then it's time for potassium to come out in order to restore the original settings. And this is called the after hyperpolarization phase. Voltage-gated potassium channels are still open, and that is why the cell surpasses -70, going into after hyperpolarization phase. There are just two terms on this slide which need further explanation. One is the absolute refractory period, and the other one is relative refractory period. What is meant by this? Obviously, green and this beige colored section are the absolute refractory period. So during this time, any additional stimulus being applied or given to this cell would not result in any alteration of what the cell is already doing. It'll complete its action potential it started and will be absolutely indifferent to any other stimuli received. But in a relative refractory period, referring to this beige colored part of the diagram, beige orangish, basically it is this segment where voltage gated potassium channels are still open, sodium channels are already in a resting state, so the cell during the relative refractory period would be possible to stimulate it. However, the stimulus has to be stronger in order to overcome whatever the difference in the cellular resting membrane gradient is from -70 to where the cell currently is. So if we needed here -70 to -55, just 15 millivolt worth stimulus, if the cell here is at -75, in order to reach 55, we would need something to change its polarity by 20 millivolts, not just 15. So essentially, that is what physiology of action potential is. Just to understand, cellular membrane, integrated protein channels, ion channels being open, closed, electrical or chemical stimulus to these channels has been discussed, or physical. Once they start opening and the cell reaches the threshold, basically it all becomes the same predictable event, just as described in this diagram, that we refer to as action potential. Should we need a little bit more information here, we can practically see extracellular, intracellular fluid, again, the same. We have a diagram, part of the cellular membrane, but this time with sodium and potassium channel being illustrated simultaneously. Picture one, resting state. All voltage, sodium and potassium channels are closed. So no movement of sodium, no movement of potassium whatsoever. And regardless how much time we measure, our resting membrane potential will be at -70 millivolts for good. Depolarizing phase is something we already talked about. We want stimulus to be applied and to start changing polarity of the cellular membrane, hopefully bringing it to the level of threshold. It may or may not happen, but it is still the depolarization phase or depolarizing phase. Depolarization to threshold starts by opening of some sodium channels. Sodium, as you can see, keeps getting into the cell. So if the cell was measured -70 with each positively charged particle, we are offsetting that kind of negativity, so to say, and we're bringing it from -70 to 69, 68, so on and so on, until we reach -55, which is threshold level. You realize that potassium channel stays firmly closed, no changes, but as the sodium channels are open, basically they bring the cell closer to threshold level, which result in rapid electrically gated channels for sodium. getting open, and massive sodium influx happens within the cell. That's why the polarity changes so quickly, goes from -55 threshold over 0 plus 30, and we are seeing this as completion of depolarization phase. Now it's time to start going down. We need to think about how the cell will repolarize. Keep in mind, it already accepted and started hosting countless number of sodium positively charged particles. So the repolarization, when a cell reaches the peak of its depolarization around plus 30 millivolts, sodium channels become closed, no more sodium ions to keep coming into the cell, but potassium channels are now open, and we're seeing the massive outflow of positively charged potassium ions back from inside of the cell into extracellular compartment. Potassium, with a high concentration of it inside the cell, low outside, simply utilizes its own diffusion gradient. So for every positive particle that came in, now we're seeing another group of positively charged ions coming out and changing and reversing membrane potential back to where we started. As the repolarization continues, potassium outflow restores the resting membrane potential. Sodium channels inactivation gates open, return to the resting state when potassium gates are closed. And then we're back to the beginning of this diagram that we can go perpetually over and over as it explains the principles of resting cellular membrane, resting membrane potential, depolarization, repolarization, and back through the same predictable cycle again and again and again if we need it. So if we generated action potential, we should not forget it has been determined at so-called trigger zone at the hillock. And from that point on, basically the signal, the current, will be sent along the axon, not the other way around. Because keep in mind, it's the electricity, and the body of the cell is already kind of changing its polarity quite significantly with sodium, potassium influx and outflux, that we cannot see electrical current generated by extra sodium channels being open, reversing backwards into direction of cellular body. It can only go along a non-polarized part, which is axon. So it can go through two different possible scenarios. For axons that do not have myelin coat, electrical signal propagates practically in a linear direction, and we call this continuous conduction. Current area, which has been depolarized, is in a green colored box, and we know that the electrical changes affecting this particular part of the membrane start slowly opening more sodium channels further distal, downward the axon. When more of them are open, basically this segment will become depolarized and will start moving downward, but it's like a continuous process which really requires a little bit more time to conduct action potential from where it was generated all the way to the axon terminal. We can say this is like disadvantage, but we have a number of different neurons where the speed is not of the utmost importance. Speed is still remarkable. Even the slowest neurons would carry action potentials at a speed between 0.5 to 2 meters per second. The fastest ones will take it up to 120 meters per second. So right away, you can see that there is a significant difference. But there are different parts of the body which really require different response time. So for example, signals about pain. Let's say you touch the hot surface and you feel extra heat creating a blister. So you're momentarily aware that you have touched the surface of a hot stove. And right away, your response will be to remove the finger from hot surface before you continue making even more profound damage. So what prompted your attention? It's a little bit of a pain that we have received. It didn't take over a second for us to remove the finger, and we're fine with that. Another example also involving pain, let's say you start feeling a little bit of a headache. Does it really make in any of our lives that headache has been detected now or a millisecond later or half a second later? We wouldn't have even time to think about what happened, or if we were to take some kind of, let's say, medication for a headache, we wouldn't even be able to leave the chair where we were comfortably seated in order to get the medication. So clearly, it's not so essential that all the signals are transmitted at the same speed. But let's give another example where response time needs to be really like minimal. You walk, you do not necessarily see where exactly you planted your foot, but you planted it on the ice patch. Momentarily, you don't have support, your foot started slipping forward, you're just about to lose balance and to experience fall, but somehow miraculously with the different parts of the body, we offset this unpleasant scenario, we preserve the balance, we don't experience the fall. That kind of response really required the fastest possible response time. And that is where signals traveling at speed of up to 120 millimeters per second are absolutely a must. So the right side of the screen explains where the principal difference in conduction of signal comes from between non-myelinated and myelinated neurons. So we have here illustration of multiple Schwann cells, considered this being a neuron of the peripheral nervous system. As we know, Schwann cell myelinates about 1 millimeter of the axonal length. So between two Schwann cells, we have segment of the axon which is not myelinated, and that is what's called the node of Ranvier. So in this case, when action potential is generated at the axon hillock, there is a huge potential difference which we have between two consecutive nodes of Ranvier. So as a result of this, the area of the axon, which is covered by myelin, basically does not allow any additional changes, but at the node of Ranvier, additional sodium channels would start opening. So as a result of gradient between previous and the next node of Ranvier, we're talking about action potential, which keeps leaping. Saltare in Latin means to leap, to jump, practically jumping from one to the next node of Ranvier until we reach the axon end. Obviously, it saves the time. It ensures that we have much faster spread of the action potential, reaching effector cells. And that is the basic difference that we see as the difference between non-myelinated and myelinated neurons, continuous versus saltatory conduction, that heavily influences the speed of transmission of action potentials. So what happens at the end? At the end, we would have action potential reaching the end, axon end, synaptic end bulb of a presynaptic neuron. If we don't have anything else happening at that point at the synaptic end bulb, basically we would have to ask ourselves a question. Why was action potential generated? Because it would not have any further effect other than kind of depolarizes and repolarizes one single cell, but not to keep moving on. Well, we know it's not truth. And we have here two neurons one with its axon end, synaptic end bulb, that is the presynaptic neuron. And at the lower part of the screen, we have another neuron diligently waiting for something to happen to it, and that is the postsynaptic neuron. As you can see, a tiny gap, which is called the synaptic cleft, it is less than micrometer, but it is still cleft. There is no physical contact between pre and postsynaptic cell. They could communicate to each other only by using some of these neurotransmitting molecules. And the purpose of this diagram is to explain how that happens. So when a nerve signal reaches, regardless whether it's continuous or saltatory conduction, the end of the axon at its synaptic end, basically it doesn't have anywhere else to go. But the electrical current, which was brought to synaptic end bulb, essentially would be used to open up voltage-gated calcium channels. Keep in mind, this is the first opportunity for us to talk about calcium ions rather than just discuss only what sodium and potassium are doing. Obviously, the membrane responds to this electrical change. Electrically gated or voltage gated calcium channels open up, and the calcium enters the cell. Inside the cell, we see those transportation vesicles containing chemically active substance that we call the neurotransmitter. Of course, the vesicles are there to keep it stored and to wait until the opportunistic moment comes. Obviously, without calcium ions, those transport vesicles containing neurotransmitter molecules would be indifferent. With presence of calcium, as indicated with number 3 on the diagram in the middle, it will move some of the transportation vesicles towards cellular membrane of the synaptic end bulb. They would, like many other transportation vesicles, just blend into the membrane of the synaptic end bulb, open up, and through the process of exocytosis, they would release the neurotransmitter substances into synaptic cleft. What makes them move from pre to postsynaptic cell? It's again plain diffusion gradient. You see how many of the neurotransmitter molecules are here and how few of them are next to the postsynaptic cell. So diffusion gradient allows movement of neurotransmitter from pre to postsynaptic cell. And here we have at the postsynaptic cell proteins that are not electrically gated, proteins that are chemically gated or ligand gated. Each of these protein channels have receptors on its extracellular fluid compartment side, ready to bind the neurotransmitter. And as the neurotransmitter freely diffuses, it's just a matter of time when these vacant receptor sites will turn into being occupied and neurotransmitter molecules will be bonded onto the surface of this protein channel. As a result of binding a neurotransmitter, these protein channels are obviously ligand-gated channels. They would open, and abundance of sodium from the extracellular fluid compartment starts moving into the cell, creating localized change in the postsynaptic cell membrane. This is a little extra that I'm asking you to take into consideration. Some sodium came in and it created a change practically only on this part of the dendrite. Other dendrites, rest of the postsynaptic neuron, is still very indifferent in a resting membrane potential. Obviously, this could be the starting moment that eventually will lead to postsynaptic neuron generating its own action potential. But we have to wait until this change practically becomes like a wave, which starts opening more of the electrically gated channels to allow even more sodium to come in. And of course, then to see quicker change in the membrane potential, reaching the threshold. And from the threshold, we know what's going to happen. Depolarization of the cell, repolarization, but it really means that the postsynaptic cell generated its own action potential and sent it along its own axon. So as the local area of this part of the dendrite changes, it builds the postsynaptic potential, which eventually, with more sodium ion coming in, more electrically gated sodium channels being open, would result in formation of another nerve impulse generated by the postsynaptic neuron. And of course, then it will send the electrical charge either to a muscle or to a gland, stimulating them to do their function. Now one can ask, what's happening between multiple neurons? Is this always very simple, or it could be made a little bit more complex? It could be made a little bit more complex because clearly through the multiple dendrites that more or less each neuron exhibits, it is immediately making it possible that on two different dendrites, two different presynaptic neurons would start influencing postsynaptic neuron. So regardless of what neurons A and B are doing, if they are stimulating only this dendrite, neuron B, or only this dendrite, neuron A, basically we're going to just see and record minor change in the resting membrane potential. Not enough to bring us to threshold level -55. If A and B neurons are stimulating at a different time, basically they would just record this kind of very minute change. But it's just a hypothetical idea for us to understand what's happening. Obviously, as we would have many presynaptic neurons, so we would have to add here neuron D and neuron E and neuron F that eventually would all want to influence the postsynaptic neuron. So when many of them are active, they could quite quickly charge and result in a formation of new action potential. This is what is called the spatial summation. Spatial space, obviously, it is multiple neurons being stimulating postsynaptic neurons simultaneously at once. They're coming from a different space, different direction. So hence we say this is the spatial summation that would result if the threshold has been reached with successfully generated action potential. Another possibility is temporal summation. I'm not sure if you know the meaning of the term tempo. In Latin, translates into English as time. So temporal summation between presynaptic and postsynaptic neurons. So let's say this presynaptic neuron just fires randomly at this synapse, and then 10 milliseconds later fires again, and then 10 milliseconds it fires again. We're going to have the series of these slight elevations, but none of them being sufficient to reach the threshold because they were apart by time. 10 milliseconds, 10 milliseconds, and 10 milliseconds later, nothing is going to really affect the postsynaptic neuron to generate action potential on its own. But if the presynaptic neuron, because it has been properly stimulated, keeps firing its own action potentials multiple times, now you see it's not every 10 or 20 milliseconds, but rather every 3-5 milliseconds, the resting membrane potential of the postsynaptic neuron will start rising through the threshold, would not have enough time to go back to resting membrane. So we're going to experience gradual climb, almost like climbing a flight of stairs, until we reach the level of -55, the threshold. And at that point, we know that we are successfully stimulating the postsynaptic neuron to fire its action potential. So it is sometimes to explain what's happening between pre and postsynaptic neurons, because it's not really black and white. I want to introduce here one useful term, easy to remember. Action potential is really all or none principle. Either it's going to happen or it won't happen. There isn't anything in between. Pure binary code. 1, 0. Happens, doesn't happen. And another good thing to know, regardless of the strength of a stimulus which has been applied to postsynaptic neuron, basically its response is always going to be the same. We would not have a situation where some exceptionally strong stimulus will force the depolarization to go to, let's say, +100 millivolts and then down to -70. It would always stop at around +30 millivolts, regardless of strength of the impulse or signal being received. So that makes it completely predictable, and as such has found its appropriate space in the physiology of nervous system. Certainly, neurons, as they tend to create a variety of different circuits, are not so easy to illustrate. So here we have just some ideas for us to broaden our horizon, really not to learn this with any kind of specific information. We can see here something that's almost like a geometric progression. One neuron stimulating other two, they are now, as they are stimulated, the postsynaptic neurons to this one, but they immediately become the presynaptic neurons to this group of 4. So 2 would stimulate 4, 4 would go to 18. And we know that one simple signal could start spreading out very quickly and involves multiple neurons in the response as a postsynaptic nature is applied to their names. That is diverging circuit. Here is the opposite, converging circuit. Multiple neurons all fire in the direction of single postsynaptic neuron, which will create action potential of its own. But this type of circuit obviously funnels it down into converging circuit. Unfortunate thing, if this neuron will become damaged, out of function, basically the entire circuitry will fail, because these 3 would still continue trying to stimulate injured, damaged, or non-existing postsynaptic neuron. Reverberating circuit, musicians would love this word, but we know what is to reverberate, just to have it continuously going. So we can see here signal generated by this cell stimulates this cell, which stimulates this cell, and its axonal sideways directed branch stimulates another parallel position neuron with its axon that stimulate neuron number 2. So this is likely to keep going for quite some time. Or on the far right side, we have a parallel after discharge circuit, practically just using multiple collaterals, like 3 different regional roads converging onto the same highway. And that is kind of example that I can use without really trying to overemphasize the value of a variety of different circuitries. I just wanted to give you some form of idea. Also, as we know, nerve cells are in the G0 phase. That means they are unable to follow the principles of mitosis and to keep multiplying throughout our life. So when a neuron is functional, it's going to look like this. When some kind of damage is done to it, and if the damage doesn't involve damaging the cellular body, that would require us to say that the damage is beyond repair, but the damage was only affecting the axon, which is quite likely like injuries of peripheral nerve are not really injuries to cellular bodies, but rather they're very long axons. So what happens to a damaged axon? It undergoes chromatolysis and so-called Wallerian degeneration. But the good point is that axon does have a chance to show its regrowth. And if it regrows into its own regeneration tube. Rate of regrowth is very slow. So practically, we can say that it is 1 millimeter per day, which is good to know for people who have sustained some, let's say, injury to their peripheral nervous system, to their nerves. And if the injury affected nerve at the elbow, when do we expect to have function of that nerve restored and we have a good quality sensation in our fingertips? Basically, distance, elbow to fingertips, might be measured as 25, 30 centimeters, depending on the length, and that is 250, 300 millimeters. So we would estimate that in order to start seeing first good signs and realistic signs of recovery, we have to be patient and allow time to pass. So in this case, we're talking about nearly a year until such a damage is showing signs of repair. We have here additional support cells, which I partly introduced in earlier section. We're going to talk about these cells like astrocytes later on when we do the physiology and anatomy of central nervous system. Right now, you can just remember its name. It's the astrocyte, astro because it looks like star, aster, and cyte because people thought for a long time that it's a neuron, but it's not. It's really like supporting cell, generating blood-brain barrier, so practically making sure that nervous tissue is protected, that substances carried by blood and released through the capillaries do not always reach the nervous tissue, which is great protection barrier, but at the same time, when people require medications, basically it becomes the obstacle how to safely deliver sufficient amount of drug needed for treatment of a number of different neurological disorders. Oligodendrocyte we mentioned as the cell which myelinates neurons of the central nervous system. We would have the next type of cell, which is called the microglial cell. Basically, it is the one which takes the role of cleaning services, practically handling remains of dead cells, cellular debris, or microorganisms if they would ever be finding access into central nervous system. Microglial cells will be ready for them to engulf them and to eliminate them before doing more harm. These cells will remind you of epithelial cells, clearly showing a little bit more of a columnar nature, even with cilia. Basically, they're called ependymal cells, and they're not really epithelium. They're also supporting cells of the central nervous system, where we have need to line some chambers of the brain called ventricles. There are 4 of them, and to allow these cells to produce cerebrospinal fluid. So that is the only type of cell within the central nervous system capable of doing this task. And of course, as we talked earlier, cells that myelinate, this is the Schwann cell, which just started interacting with multiple axons. Of course, this is not complete process of myelination. When it's properly done, the Schwann cell has to create multiple layers of its own lipid product around the axon to completely wrap it up and to make this electrical insulator. That's why we have nodes of NVA as being exposed parts of the axon, enabling electrical spark to jump from one node of NVA to another one. And also support cell that is called a satellite cell, Very important because neurons, as we know, are extremely sensitive, especially those ones that would be seen as part of the peripheral nervous system. So in order to protect them, basically the satellite cells are creating a little bit of a shell around group of neuronal bodies that form the ganglion, and with satellite cells creating a protective shield. Basically, that would ensure that multiple neurons within a ganglion would be able to survive somewhat harsh conditions of the rest of the body, whether it's thoracic cavity or abdominal pelvic space, but definitely not as good as protected as brain within the skull or spinal cord within the spinal canal. And one diagram which doesn't really bring anything new to us. However, it shows its entire cytoplasmic or plasma membrane in 3 different colors: blue, green, and red. Maybe it's not high achievement type of diagram, but perhaps might help you understand and memorize the action potential physiology a little bit faster. Plasma membrane, colored blue, includes channels that are chemically gated. Okay, that's great. That is idea. Receptor and neurotransmitter bonding together, protein channels, chemically gated, keep opening up, keep changing resting membrane potential of this cell locally, but all the sum of these charges practically becomes more and more abundant. And as a result of that, it will activate some additional sodium voltage gated channels, so the change would go much faster. That's why I would prefer that there is a hint of green also in between just blue, but it's a schematic diagram, so don't worry so much about it. The green colored part, obviously axon, plasma membrane, that would include voltage gated channels of both sodium and potassium. So that could be information that we use to explain quicker what is the saltatory conduction. And here at the very end, at the synaptic end bulbs, first time ever, we noticed that there are some other channels, voltage-gated calcium channels. We needed calcium to come into synaptic end bulb to stimulate movement of vesicles containing neurotransmitter, and as such, practically to become that kind of triggering point for successful completion of synaptic connection between 2 neurons. As you can see here, the black screen is usually a sign that slideshow comes to its end. And as a result of that, I would just say that this also becomes the end of this recording. So we have created together another section. And of course, we are also to add a little bit of extra information for muscle tissue, as we would need to take a look into individual myofibril to understand what the sarcomere is, and to then further work our way through what is called the sliding filament theory, which explains how and why the muscle contracts, and then neuromuscular junction, explaining this kind of nice interaction and integration between nervous system, stimulating the muscle tissue to do contraction. So I hope that you like this presentation, and hope to see you soon again at the Anatomy Made Simple by Dr. T. Next video will be about sliding filament theory, muscle contraction. Until then, enjoy the rest of this day. Thank you very much.